will review the anatomy and function of the juxtaglomerular apparatus. The renin-angiotensin system, RAS, located in the kidneys, is the primary regulatory system connecting blood pressure with the production of aldosterone. A vital sensor in the release of aldosterone is a specific cell in the kidney known as the juxtaglomerular cell. Positioned near the glomerulus of the renal nephron, these cells react to drops in renal arterial blood pressure. Juxtaglomerular cells are part of the afferent arteriolar wall, the blood vessel that carries blood to the glomerular capillaries in Bowman's capsule. Each adult human kidney comprises about 1 million nephrons. Each nephron features a Bowman's capsule, a filtering structure containing capillaries through which blood circulates and is purified. Blood enters these capillaries via an afferent arteriole and exits through an efferent arteriole. In the nephron, juxtaglomerular cells are located next to specialized sodium-sensing cells at the peak of the loop of Henle's ascending limb. This region, known as the macula densa, is where these cells identify changes in the tubular fluid sodium ion concentration. The juxtaglomerular cells respond to changes in renal arterial blood pressure. They are also innervated by sympathetic nerve fibers. In the event of a drop in blood pressure, these cells react directly, and the sympathetic nerve fiber activity intensifies, further stimulating the juxtaglomerular cells. The collection of juxtaglomerular cells, macula densa cells present in the distal convoluted tubule, and the glomerular afferent arteriole are often referred to as the juxtaglomerular apparatus. When triggered, the juxtaglomerular cells release a protease enzyme called renin into the bloodstream. This enzyme acts on a plasma protein known as angiotensinogen, synthesized in the liver. From this interaction, a decapeptide named angiotensin 1 is created. This peptide is then processed by another enzyme, the angiotensin-converting enzyme, which trims down 2 amino acids, resulting in an octapeptide called angiotensin 2. This sequence of events, starting from the renal release of renin to the final production of angiotensin 2, is what constitutes the renin-angiotensin system. Angiotensin 2 is also used to produce a septipeptide, angiotensin 3, the physiological significance of which remains uncertain. The decrease in the sodium ion concentration in the glomerular filtrate as it reaches the top of the loop of Henle's ascending limb is another trigger for renin production from the juxtaglomerular cells. This decrease is detected by the specialized macula densa cells, which then communicate with the adjacent juxtaglomerular cells. Angiotensin 2 is a potent pressor molecule that increases renal proximal tubular sodium reabsorption and stimulates the adrenal cortex's zona glomerulosa cells to synthesize aldosterone. It can bind to G-protein-coupled angiotensin 2 receptors, AT1 receptor and AT2 receptor, present in various tissues, including the brain's neurons, cardiac and vascular smooth muscle, and the adrenal cortex's glomerulosa cells. The renin-angiotensin-aldosterone system often refers to the entire regulatory pathway, with angiotensin 2 being the principal stimulus for aldosterone production. Aldosterone has a chronic pressor effect, making the renin-angiotensin-aldosterone system an important target for antihypertensive drugs, like angiotensin-converting enzyme inhibitors, which are often used in hypertension treatment. Stay tuned for more fascinating insights into human physiology on our channel. Don't forget to like, share, and subscribe.